This is a presentation about tissue culture for GCSE biology. So what is tissue culture? What are the advantages of using tissue culture in plant breeding? What are the advantages of using tissue culture in medical research? This is under Marion Bayen. He's a cancer sufferer, or he was a cancer sufferer, until they transplanted a trachea. But he wasn't an ordinary trachea. In 2011, he was the first person to receive a tissue-engineered synthetic organ. This means they made a trachea for him. They made it out of stem cells from his bone marrow, which matched his tissue type, and therefore would have avoided any complications with rejection. But how did they make it? Well, they used a technique called tissue culturing. So what is tissue culturing? Tissue culturing is growing cells or tissues on a liquid or solid medium, a nutrient agar. This agar contains all the things that are required for things like respiration. Many cells can be grown and all of them are identical. We refer to a callus as a clump or a group of undifferentiated cells. Here's a picture of a callus. These can be treated to cause specialization or differentiation later. What are the advantages of tissue culturing? We can prevent the extinction of rare species. Well, not those species. This is usually applied to plants. We can produce individuals that are difficult to grow from seeds, for example orchids. And the technique is also used to clone GM plants. Tissue culturing can't be used to clone whole animals because animal cells are already differentiated. Plant cells, however, are meristematic. They act like stem cells and are undifferentiated. Now there are two methods to tissue culture a plant. This diagram is on page 86 and 87 of your textbook. Before you start anything, you must sterilize your plant material that you've cut off. Sterilization stops you from growing any pathogens and multiplying them to harmful levels. Sometimes, what you can do is take a piece of plant and grow it in a sterile nutrient medium. Once the plant is big enough, you treat it with hormones so that it develops roots and shoots. And once these roots and shoots are fully formed, then you can plant them into soil or compost to grow as usual. The other way you do this is you can grow yourself a callus. In growing a callus, you only take some very small amounts of the plant itself, so only a few cells, and you multiply those cells. So only a few cells are cut off, then placed into the sterile nutrient, and then it will start to grow. The callus, once it's big enough, will be treated with hormones, so it will start to develop shoots and roots. Once the shoots and roots are fully formed, again, we transfer it into the compost or the soil. The method at the top involves taking cuttings, or sometimes you might refer to them as explants. The bottom section shows you the method when you develop cell samples, and that will develop into a callus. Uses of tissue cultures in medicine. A thin layer of culture grown on a solid medium can be used to study how cells communicate with each other. Cell cultures are used to study viruses. These viruses can't replicate outside of cells. Cultures of cancer cells can be used to study how cells communicate and spread. Scientists are able to test treatment without harming animals or humans. Further uses of tissue cultures in medicine include, like in the case of Andemaria BN, some artificial tissues can be grown like his trachea. An unreactive material like glass can be crafted to act as a support for the growing tissue. Not all organs can be grown like this, but there are attempts at combining technology and cells, like this liver cell device, which allows patients to use this while their own liver cells are regenerating from damage. Like the video if you think it helped.